Alright, so the next thing we're going to talk about is fructose. So fructose is commonly known as a fruit sugar. Okay, so if you've seen anything like this, where they would refer to fructose as a fruit sugar, right? So it's a fruit sugar in that it is soluble in it is a soluble monosaccharide, and it's usually found in foods like honey, in flowers, berries, and in roots. So this is where we can find root crops, actually root crops. Alright, so the open chain of fructose has a ketone, so it's actually the opposite of a glucose. Okay, so you can say fructose is a structure isomer of glucose, because glucose has an aldehyde group but fructose has a ketone group. So when you look at this, you find that if this is fructose, okay, so double bonded to oxygen. Sorry, let me just erase this part. Alright, so we have this bond that is happening right here. Okay, so you notice that this is a ketone group. So fructose has a ketone instead of an aldehyde. So what happens is that during the ring formation in the open chain, so this is the open chain of fructose, this carbon number 2 is going to combine with carbon number 5 somewhere there, yeah. So when you look at this, this is what is going to happen. So when carbon number 2 combines with the OH group, so when this oxygen combines with the OH group on carbon number 5, it forms a 5-membered furanose ring. Okay, so let me just erase this part so that we're able to see what exactly is happening. Okay, so we have this branching out to form a 5-membered furanose ring. Okay, okay, so this is the 5-membered furanose ring. So this is a furanose ring. So I want you to notice this. When forming a ring in glucose, notice that the carbon 1 binds with carbon 5, but in this fructose, carbon 2 binds with carbon 5. So this is to tell you that for this to form a ring, the aldehyde group on carbon number 1 is what binds with carbon number 5, and in fructose, the ketone group on carbon number 2 is what binds with the OH on carbon number 5. Okay, so this is to tell you that it's the function of groups that are participating in this chemical reaction that we're able to see. Alright, so like glucose, fructose is also a source of energy and can actually also be used in cellular respiration. Okay, so we use fructose to give us energy as well. Alright, so having said that, fructose is actually a weak reducing sugar. So this is a very important point. It is a weak reducing sugar. So because it has a ketone group, and we know that the ketone is on carbon number 2, you find that this group, which is the ketone group that is on carbon number 2, when you looked at that chain we drew earlier on, okay, if this is CH2 or H, this group is somehow or somewhat hidden. Okay, it's a bit shielded by these other groups, or rather these molecules that surround it. Okay, there's a way in which these groups can actually prevent it from effectively acting as a reducing sugar. Okay, so it is a weak reducing sugar. So because of its open chain structure, it is able to isomerize into either an aldose glucose, into an aldose, which is a glucose. So they're saying it can be it's an isomer of glucose, in that it has a ketone group, but for glucose it has an aldose group. So carbon 2 of the fructose is asymmetric, leading to the formation of the beta and the alpha types of isomers. Okay, all right, so it is can also form alpha and beta isomers just like carbon. All right, another thing that we're going to talk about is the disaccharides. Okay, so when we look at these disaccharides, having explained about disaccharides, we know that they are made up of two monosaccharides. Okay, so don't forget this point. This point is very important. So these are made up of two sugars or two monosaccharides, and they are also crystalline in nature. Okay, they are crystalline. Okay, they're crystalline. They taste sweet and they readily dissolve, so they are soluble in water. Okay, so the bond that is formed is actually called a glycosidic bond. So let me just highlight this. The bond between two monosaccharide is called a glycosidic bond. Okay, so this glycosidic bond comes about due to a dehydration synthesis, or rather a condensation reaction. This is where water is released in the process of making, or rather producing, a bond. Okay, so we'll see how exactly this happens. All right, so having said that, the bond that is formed is called the glycosidic bond, and the common disaccharides that we have sucrose, we have lactose, we also have maltose. Okay, so we're going to talk about these individually, and these disaccharides are temporary energy stores that can also be hydrolyzed to give us energy during a chemical reaction. Okay, so having said that, we need to note that sorry, pardon me. All right, so we're going to look at this part while we look at the sucrose individually as a disaccharide. So we're going to look at sucrose. Okay, so when you look at sucrose, sucrose is referred to as cane sugar or sugar cane. Okay, so this sugar cane, it has two dimers. One is called fructose, the other one is called glucose. Okay, together they make a disaccharide that we call sucrose. All right, so what happens is that like we said, the functional groups are the ones that are involved in forming a bond. So this means carbon number 1, since the carbonyl on the glucose is on carbon number 1, but the carbonyl group on fructose is on carbon number 2. So what happens is that the bond is going to be called alpha 1, 2. Okay, because this is where these two sugars are interlinking. So this is to tell you that the alpha glucose, okay, the alpha glucose is going to bond with the beta fructose, all right, in order to form this bond. So it is also used in cellular respiration, and the molecular formula for sucrose is C12H22 or 11. So sucrose is, as you all know, it's abundant in plants, and is translocated through very large phloem vessels. All right. Now, something unique about sucrose is that it is a non-reducing sugar. So we're going to look at why exactly is this fructose being referred to as a non-reducing sugar. So it is non-reducing. Okay. The reason why is because when we look at fructose, let me just draw a rough sketch. This is fructose. Fructose is binding on this is carbon number 2. This is where the bond is being made. And then on glucose, which is a 6-membered furanose ring, this is carbon number 1. So the bond is being made somewhere here. This is the glycosidic bond. Okay. Now you notice that the functional groups, this aldehyde group and the ketone group, they are both involved. They're both involved in bond formation, right? They are involved in bond formation. Hence, you find that these groups are the ones that give the atom its reducing abilities. Therefore, if they are involved in a chemical reaction or rather in bond formation, they will be unable to actually reduce other elements. Okay, to reduce, we know it's to add hydrogen or to remove oxygen. So this sucrose is unable to do that because all the functional groups are involved in the bond formation. All right. So sucrose is taken up into glucose and fructose, which can be used to generate energy using tissue respiration. So this can also be used as a sweetener that can be used for baking and so on and so forth. So this point is important, the one for the bond formation. So you need to know why sucrose is not a reducing sugar. Okay, so we said that the functional groups, the aldehyde or the aldose of glucose and the ketone group of the fructose, they are both involved in bond formation, like we said. So because of this, they will not have any space through which they can even react with other substances or other molecules in nature. So this is all about sucrose. All right, the next thing we're going to talk about is lactose. Okay, so talk about lactose. Okay, so when you look at lactose is referred to as milk sugar. Right, so because it's called the milk sugar, it is joined by galactose and glucose. All right, so it is linked through a beta 1,4 glycosidic bond. So this is to tell you, when you look at this, you will know that the carbon number 1 on the beta galactose combines with carbon number 4 on alpha glucose. Hence the bond is beta 1,4 glycosidic bond. Okay, so it is important for you to know these bonds individually. So the chemical formula for galactose is C12H22O11. So lactose is also soluble in water. Okay, because it's able to form hydrogen bonds. It's soluble in water and can be extracted as well from sweet whey. So this is sweet whey, it can be extracted from it. So the formation is where we need to know that galactose is actually a 6-membered ring, so is glucose. Okay, so they're both pyranose. When you look at this, they are pyranose rings. Okay, so when we form this bond, we have lactose, where we have galactose and we have glucose. This is galactose. All right, so lactose is a reducing sugar because the carbon 1 of the glucose, so if this is carbon 1 or the other way around, the carbon 1 of the glucose is free. Okay, so this is to tell you that the aldehyde group on glucose is actually free. Alright, so because it's free, so this aldehyde group in glucose is free. Okay, so because it's free, it means it to participate in reduction chemical reactions. Okay, hence lactose is a non-reducing sugar. Alright, so this is to tell that galactose, if galactose and glucose are forming lactose, it means if you want to get back galactose and glucose, you can still break down this lactose to give you the individual subunits that are used to make it. Okay, the last disaccharide is called maltose. Okay, we have what we call maltose. Now maltose is called malt sugar. Okay, so it is water soluble. It is composed of two glucose. So it is composed of two glucose monomers. It is soluble. It is also a reducing sugar. Okay, the bond between glucose molecules is alpha 1,4. So this is to tell you that on carbon number 1 of the glucose and the carbon number 4 of another glucose, they make this glycosidic bond. Okay, so the other aldehyde group is actually left free. So because carbon number 4 is the one that is bonding to carbon number 1, it means the carbon number 1 on the other glucose molecule is actually free. Hence, it can participate in a chemical reaction. This is what they mean. So maltose is broken down and digested to give us energy in the body as mammals, right? So that's it about maltose. Maltose can also be broken down into glucose and into glucose units by the enzyme maltase. Okay, so we have an enzyme that we call maltase that actually breaks down this maltose molecule. Okay, so this is the enzyme, it's called maltase. Alright, so talk about polysaccharides. Already from its name you're able to tell. So polysaccharides. So the first thing that polysaccharides is that they are not sweet. They're not sweet. 2, they can be used for energy storage. And then they are insoluble, meaning they cannot dissolve in water, right? They do not dissolve, or they slightly dissolve in water. Alright, so polysaccharides can either be can be used for food or energy storages. Okay, another thing that we need to notice is we'll look at some examples. For instance, we have starch, glycogen, cellulose, and chitin. So look at these types of polysaccharides individually, right? Okay, so these are some of the polysaccharides that we actually have. Alright, so the first polysaccharide we'll talk about is starch. Okay, starch is a polymer of alpha glucose. So if it's a polysaccharide, it means it's a long chain of glucose units. Okay, these are glucose units that are being used to make this polymer, right? So they are formed through these glycosidic bonds that we're talking about. So it is insoluble in cold water. For instance, if we get millimil, okay, so if this is millimil, we pour it in water, you will notice that the millimil at first would just form some sort of balls on top. If we know that the water is cold, because it's insoluble in water, right? And then you find that because of this low solubility, it is very hard to break down into its single monomer. That's why it's used as a storage. Now, this starch has two components, okay? It has two components. One is amylose, another one is amylopectin. Okay, so amylose has a 1,4 glycosidic bond. Amylopectin has 1,4 stroke 1,6. So let me briefly explain about that. So when you look at amylose, right? Amylose is a long chain of glucose molecules. So it is tightly packed, okay, in a spiral manner. Another one is amylopectin. It may be packed like this, but it's got some branches, okay. So these branches, so this is the 1,4 and this one is 1,6 branch. This is amylopectin. Okay, so amylose is a straight chain of structure that consists of thousands of alpha glucose linked through alpha 1,4 glycosidic bonds. Alright, so when we put this amylose in water and test it with the iodine solution, it will give us a blue-black color. This is because the iodine solution is able to seep through this amylose, right? It is able to interact with the molecules, hence it will give a blue-black. So a blue-black solution, right? So when we look at this, the amylose actually constitutes 20 to 30% of starch. Okay, so amylose doesn't have a huge portion of starch. But when you look at amylopectin, it is similar in structure, but it has 1,6 glycosidic bond and its chains are actually shorter. So these chains that you're able to see are actually short, and the chains within its structure are also short. Okay, so a suspension of this amylopectin in water, and when it's tested with iodine, or rather the test for starch, you find that it will retain the color of iodine, which is brown. Okay, so it will then give us a red violet color with the iodine solution, because there will be no interaction between the iodide complex and this amylopectin, and this amylopectin is one that constitutes 70 to 80% of starch. Okay, this is what we're talking about. Okay, all right, so we also have glycogen. So glycogen is an animal equivalent. It is an animal equivalent of amylopectin. So when you look at glycogen, this is a food storage or energy reserve in eukaryotic cells, for instance, an animal cell. So it is an animal's equivalent. This is to tell you that it is similar to amylopectin in structure. So this would tell you that glycogen has 1,4 glycosidic bond as well as 1,6 glycosidic bonds, okay, between its monomers. So it can exist in form of granules in the cytoplasm. So it can exist in form of granules in cytoplasm, right? And glycogen it can be hydrolyzed into glucose for respiration. Many are the times when the body is in need of oxygen, glycogen can be used to supply the body with energy by breaking down those bonds, okay? So it is similar to amylopectin, but its branches are shorter. So branches are shorter and they are more frequent, meaning there are many, okay? They're more frequent. So the glucose chains of glycogen are organized in branches of a tree. So it is like a long tree of glucose molecules because it resembles amylopectin, okay? Another thing that we talk about is cellulose, okay? Alright, we have cellulose. So cellulose is water insoluble, okay, polymer with beta glucose, okay, it's got beta glucose with CH2 or H groups alternating above and below the plane of the unbranched cellulose molecules. So the hydroxyl groups which project outwards from each chain, they form the hydrogen bonds with the neighboring cellulose molecules, okay? So what we notice is that this cellulose is commonly found in plant cell walls, okay? So if we have a leaf right here, right, you find that the leaf is made up, the cell wall is made up of cellulose, okay? And this cellulose is insoluble in water and it is a polymer of glucose. So these bundles are arranged in layers. So when you look at cellulose, it has layers of bundles, okay? And when we have many layers of bundles, you find that things cannot easily seep through the cell wall. Hence, it gives the plant cell structural support. So the cellulose layers are fully permeable to water, meaning they can allow water in, which is an important function of the plant cell. So the cellulose is the most abundant. So this is to tell you that the cellulose is commonly found throughout the world. You find that cellulose is in more abundance on the scale of 1 to 10, okay? So cellulose is the most abundant. We also have what we call chitin, okay? We have what we call chitin, okay? We have chitin. So this chitin is an unbranched polymer. So if it's an unbranched, meaning it's amylose, okay? So we have N-acetyl glucosamine. So it's found in the cell wall of fungi and in the exoskeleton. So by exoskeleton we mean the skeleton of certain insects or animals rather is found on the outside instead on the inside. So some of them are made out of chitin. In fact, most of them are made out of chitin, and it is similar to cellulose, okay? The only difference is that is that the hydroxyl group, the OH group on carbon number 2 of each glucose unit has been replaced by acimido. So in the cellulose we have an OH group on carbon number 2, but in chitin on carbon number 2 we have a molecule that we call acimido, which is NH double bond O CH3. Okay, so this is chitin. So remember, it is used in the exoskeleton of certain insects, the crabs, we have shrimp, and it is very similar to the cellulose, except on carbon number 2 cellulose has OH. On carbon number 2 in chitin it has acimido, the acimido group. Okay, so this is the end of our carbohydrates video. Hope you enjoyed.